Hey guys, welcome to video number 6. This is talking about cell organelles. So this is no longer in Module 10, but this is something that you're going to need to know for future science classes. So we're going to go ahead and just combine it with Module 10. So the first type of cell that we're going to discuss is prokaryote cells, or also known as prokaryotic. And that's just one of the main types of cells. The other type would be eukaryote or eukaryotic. So with this characteristics, prokaryotes make up bacteria organisms, which we know, of course, is in the Monera kingdom. And most of the time, these organisms are unicellular, which means that they are literally made up of one prokaryote cell. That's how simple they are. Now, the organelles that are in most prokaryote cells. So keep in mind that not every single prokaryote cell is going to have these organelles. This is just a basic list that is in most of them. But there's always going to be some cells that do not fit this profile. So I'm just going to go through and kind of just mention a little bit about each of these, and then I'll show you a picture when we get finished with this. So bacteria cells have 3 layers of protection. Now let's think about why they would need 3 layers of protection. Well, if you think about it, if the entire organism has one cell, that means that they get one shot at staying alive. Now, think about humans. Humans, we are made up of lots of cells that do a lot of different things. So our cells don't necessarily need to be protected as much, because our cells, they can die, but then they can make new cells just as quickly. So we don't need all of those layers of protection like bacteria organisms do. So the first layer of protection is the outermost layer, and that's the capsule. It is kind of like a hard shell that protects the gushy stuff inside. The second layer of protection is the cell wall. And then the third layer of protection is the cell membrane. And us eukaryote organisms, we have mainly just a cell membrane to protect us. Now, the cytoplasm, we talked about this in Module 9 as well. The cytoplasm is this gooey stuff inside that holds everything in place. Okay, you can kind of think of it as the blood of the cell. Ribosomes, so sorry about that, guys. This is my cat. Get down, Panther. So ribosomes, these little guys are really important. And in prokaryote cells, these ribosomes just kind of float around randomly, but they make proteins. And you're not going to fully understand this yet, but proteins are really important to make new cells and new DNA. Now keep in mind, DNA is extremely important in order to make a new cell because it's the instruction book. But without the DNA, we can't make a new cell. Well, without proteins, we don't have DNA. So these ribosomes are really important when they make the proteins because it all plays a role with DNA. Okay, let's move on to the next page. So that leads us to DNA and plasmid. So DNA is this tangled up mess that's usually in the center of a prokaryote cell. And I have told y'all this lots of times. It is the instructions to build a new cell. And all of God's living creatures has its own personal instruction book. So we all have different DNAs, which is just really amazing to think about. Now the plasmid is also a type of DNA, except it is circular. So in the picture that I'm getting ready to show you, you'll see just normal DNA, and you'll see plasmid DNA. Now, all you really need to know about the plasmid is that it's circular DNA. When you get to high school, you will learn lots more about plasmid, but let's just say that it is really, really handy for scientific technology, and we'll just leave it at that. But you'll get to learn that in biology class. The flagellum, notice what I put in parentheses here, in some cells, not all. Okay, this is a tail used for swimming. So bacteria is really good at moving around. Some of them have tails for swimming. Some of them have cilia or pili. Some of them also have pseudopods for essentially walking. So back to the flagellum though, it is used for swimming. Cilia and pili, these are similar, but they are hair-like little structures that's on the outside of the cell, and they can help cells move because they can kind of swish around. So if a cell is in a liquid type substance, then it can help the cell swim through the liquid. But it's also used to provide outer senses. That way the cell knows exactly what is surrounding it. Remember the eyelash example that I told you about earlier? So the eyelashes on our eyes allows us to sense when something is near our eyeball. So then when our eyelashes get touched by something, it automatically makes us want to shut our eye for protection. So the cilia and this pili are kind of like eyelashes. It allows the cell to know what's going around as it floats through the environment. Eye spots. Eye spots are really cool. This is something that some bacteria cells have, but we also talked about this with protista. Some protista have this as well, but it allows bacteria that is photosynthetic to actually find sunlight. Contractile vacuoles, again, these are in some cells, not all, but the vacuole itself stores water. So this contractile vacuole, it can expand and contract, expand and contract. So depending on how much water needs to be inside of the cell, it can either remove water or take in water. Pseudopods represent fake feet. So pseudo is fake, pod is feet. So fake feet that allow cells to move. And again, it's kind of like walking, but it doesn't really look like our typical view of walking. It's more of a jiggling motion. When the cell decides to move, it kind of jiggles along the surface that it needs to move along. So it's just kind of neat to see, though, if you were able to see that underneath a microscope. Now, here is the picture of a typical prokaryote cell. Again, they all look different, but this is just a basic picture. And there's just a few things that I want to mention. So the plasma membrane, this is also called the cell membrane. It's just two different names for it. So if you notice, the cell membrane is this green layer right here. Okay, so it is the innermost layer of protection. The cell wall is the second layer of protection, so it's this yellow layer. And then the capsule, let's see. Yep, capsule is right beside of it. So capsule is this red layer. It's the outermost layer of protection. So 3 layers of protection. Now this jumbled up mess right here in the center, this blue stuff, is DNA. Also called nucleoid, but you only need to know that it is DNA. And it is free floating in the cell. It does not have a nucleus. Now, the circular blue pieces, those are the famous plasmids. So those are also DNA. They are just in a circular shape. And again, like I mentioned before, they are really useful with scientific technology. Now the pili, You can also put beside pili /cilia. Okay, pili or pilus is the singular version of that. And cilia, they kind of do the same thing to where they can either help the cell move or they can just act as sort of like little outside receptors so the cell knows what it is near. And then cytoplasm is all of this background stuff. It's like a light blue color that holds everything in place. And then these little red dots all over the place are ribosomes, which make the proteins. And like I mentioned, the proteins are really important for the DNA to be made. And then this tail is the flagellum that allows the cell to swim as it needs to. And rather than doing some study guide questions today, I'm actually going to wait to do the cell organelle study guide questions until we've talked about eukaryotic cells. So this will conclude our video number 6.